Cell division consists of two processes, mitosis and cytokinesis. Mitosis, division of the nucleus and its chromosomes, is divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis is followed by cytokinesis, when the cytoplasm splits to form 2 separate daughter cells. The cytoplasm of a cell in late interphase contains 2 centrosomes, each of which may contain a pair of centrioles. In the nucleus, the chromosomes have been replicated during S phase, but are still dispersed in the form of chromatin. 1 or more nucleoli are present. During prophase, the nucleoli disappear and chromatin fibers coil up to become discrete chromosomes. Each chromosome consists of 2 identical sister chromatids, joined at the centromere. Microtubules grow out from the centrosomes, initiating formation of the mitotic spindle. During prometaphase, the nuclear envelope breaks into fragments. Some of the spindle fibers reach the chromosomes and attach to kinetochores, structures made of proteins and specific sections of DNA at the centromeres. Non-kinetochore microtubules overlap with those coming from the opposite pole. At metaphase, the mitotic spindle is fully formed, and the microtubules attached to kinetochores move the chromosomes to the metaphase plate, an imaginary plane equidistant from the poles. Anaphase begins when the 2 centromeres of each chromosome come apart, separating the sister chromatids. Once separate, each sister chromatid is considered a full-fledged daughter chromosome. Motor proteins of the kinetochores walk the daughter chromosomes along the spindle microtubules toward opposite poles, and the microtubules shorten. At the same time, the spindle microtubules not attached to chromosomes lengthen, pushing the 2 poles farther apart and elongating the cell. During telophase, nuclear envelopes form around the identical sets of chromosomes at the 2 poles of the cell. The chromosomes uncoil and nucleoli appear in the 2 new nuclei. Meanwhile, cytokinesis begins, splitting the cytoplasm and separating the 2 daughter cells. In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in 2, creating 2 identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the 2 daughter cells. The cell plate's contents join the parental cell wall. The result is 2 daughter cells, each bounded by its own continuous plasma membrane and cell wall.